All right, hey guys. I thought I would just show you here for your primer design challenge, sort of a video walkthrough of how I would go about doing this, and maybe a protocol that you could write out or follow, since this is your first time trying to do something like design primers. So there's a few things that I would have pulled up, and the very first thing that I would have is the paper to get through some of the experimental design stuff, and then really kind of one important detail. That will just shorten this up a little bit for you. So we know that the paper is broken up into several different spots here, with abstract kind of being first, and you got a title. Where I'm actually going to have you go is to scroll through and go down to the methods right here. So you'll notice in the methods we have an experimental strategy where they're talking about, well, here's the way that we design things, right? And we're interested specifically in looking at the TAS2R38 gene, and that is the gene that codes for the protein that is the bitter taste receptor, right? So we've got two things that I want to point out here, and they're these things called accession numbers. Now, accession numbers are numbers that you can plug into a database like NCBI, which are these weird looking numbers right here that will send you directly to a sequence or some sort of gene of interest. So here for AY258597, right, and then you've got one that follows right after that, it gives you the PTC non-taster and the taster alleles respectively. So let's say that I wanted to go look at the sequence for a taster. Well, what I would do is I would write this down, or in my case, because I've got a digital copy of this, I'm just going to go ahead and copy that accession number. So the next thing that you're going to want to do is go ahead and pull up NCBI, get rid of that paper. And we're going to go to NCBI, right? So you can see there's the URL right there. You can just search for it somewhere and this thing will pop up. And what I'm specifically interested in is getting the nucleotide sequence. So I'm going to go right over here and change this where I've got a huge list of options of types of databases that I can search through if I have the right information. And I'm going to go down to nucleotide. I'm going to click on that, right, because that's what I'm interested in. And then I'm just going to paste or type my accession number into there. Now, every single gene that gets mapped or sequence that gets mapped gets an accession number. So it just helps you really easily find the exact sequence that you want if you have the accession number. Right, so I'm just going to go ahead and click on that. And you guys see this gets pulled up here where you've got a title. And then if you scroll down, you've got a lot of other information here where it's giving you like some organism information. Here you've got some gene information, some mRNA, the coding sequence, and then this is the thing that you're actually interested in, which is the sequence of nucleotides. Now you can just go ahead and grab that with your cursor and highlight all 1002 bases and just hit copy. And then this is the thing that you're going to want to keep and put either into SnapGene if that's on the desktop, or you can just drop that into a Google Doc or a Word doc. But this is the information that we want, right? Because this is actually going to be the gene that codes for the non-taster allele. Okay, so now that I've got all that information, I've got it copied, I'm going to go ahead and open up SnapGene here, and I'm going to get a new file. And I'm just going to paste in all my A's, C's, T's, and G's, right? So I've got that whole thing. In topology under here, it's going to ask if this is a circular or a linear sequence. This is gDNA, so this is going to be linear. And then I'm just going to hit OK. Right? And then what pops up is this sequence that's just the non-taster allele. Down here you can see there's a map, which is what we're currently looking at. There's a sequence. There's where restriction enzyme sites are. I'm going to go ahead and click on sequence down here. And then up pops all this information that I need. Right? So it's got a bunch of, you guys can see this. This is where some restriction enzymes are going to cut within the sequence. This is the end, and then this is just the start. Right? So what we're particularly interested in is in that first single nucleotide polymorphism, which is somewhere around in that range. But just to double check, I'm going to pull the paper back up, and I'm going to scroll down to one of these figures. Right? And we've got this sequence is where our SNP is at. So I'm going to look a little bit upstream of that. And I've got a sequence here that I'm going to write down just on a piece of paper. Right, so I can go ahead and search that on SnapGene. So I'm going to write down TGA, right? So this portion, AGA, GG. right? So I just have that written down on a piece of notebook paper. And I'm going to go back into SnapGene here, and I'm just going to search that. So I just hit Control F, and now I can search for that sequence. And there it is. So there it pops up. Right? So what we're looking at is this region right over here that we want to make sure that we amplify. Just this region here, right? Because that's where the polymorphism is at. So I'm going to start designing way upstream of that sequence up here. And then my reverse primer is going to be kind of down here, and that's so I bracket my sequence of interest, right? Now, something I do want to point out to you is when you actually get this sequenced, which is why we're doing this, right? When they actually send you back what your sequence looks like, you're going to get this weird kind of, I'm going to pull one up real quick. You're going to get this weird code that's sent from them that looks something like this, right? So this is what Sanger sequencing looks like when it's actually sent to you by a company. And what you should notice is all this noise, right? So I'm actually going to flip this and go over here, right? So look at the start of this. So when you've got all of this beginning portion of a sequence, you can see this is N, which stands for just any nucleotide. So when the computer and their sequencing technology is actually going through and trying to sequence the data, the first 100 bases or so from your primer, downstream of your primer, are kind of junk. So we want to make sure that we give ourselves plenty of space so that by the time we actually get to the portion that we want read really accurately, we're getting it, right? So you can see as we go further and further along how much cleaner the read gets. So you can see the height of these peaks right here are giving you an idea of how good the read is. And as you go further and further and further, you get these really clean reads and you get all these letters. But your first 100 bases or so that come after your primer are going to be kind of junk. So we want to give ourselves a buffer region. So in English, like what I'm saying is, well, if this is our region, I don't want to start right there with primer design. I want to start way back here somewhere, because all of this is going to get read out really noisy, and we're not going to get something that's a clean sequence. So I'm going to go and probably start designing somewhere in this region for my forward primer, and then the reverse primer just needs to be bracketed and downstream somewhere over here. So if I start grabbing nucleotides and just dragging, you guys can see there's a melting temperature there in purple, right? So I'm going to grab somewhere around 20 bases until I'm around 60 degrees. And that's usually where I'll design my primers at. And I'm just going to stop right there. Right, so this would be an example right up here for the 5-3 of a forward primer. Right, so now what I want to do is just check my work to make sure that that's okay. So I'm going to hit copy. And you can do this in a Word or a Google document also. I would just grab the first maybe 20 nucleotides, and then you're going to want to check, right? So if SnapGene isn't on the computer, or you didn't have access to it, it's okay. That's still going to work, right? You're still functionally pretty much doing the same thing. Now on the Google Doc that's on Classroom, you can go to this link, and it's called a LEGO Calc, and what it's going to do is calculate how good our primer is, right? So it's going to give us lots of important information. So there's the primer that I just copied, and I said, well, I think that's going to be a good one, right? And then I'm just going to hit right down here, calculate. Now where you want to look is for some important constraints, right? One is the length, and we know that we want our primers to be anywhere in between 18 and maybe 24 base pairs in length. I want my GC content to be in between, I mean, ideally it's going to be in between 50% and 60%. Right here I've got 57%, so I'm pretty comfortable with that. And then I'm looking over here at the melting temperature. Now with melting temperature, I'd really like this thing when I'm designing primers. I'd really like this thing to be around 60 degrees. My target is 60 degrees. The reason I want it to be 60 degrees is for when I program the thermocycler, my annealing temperature I'd like to be at about 55. Now that's getting into like some deep detail, but really what you need to know is this one, the basic, what I'm trying to target is 60 degrees when I'm checking my primers to see if they're going to be good or not, right? So I'd like this to go up a little bit. So the way that I make my melting temperature go up is I just add more bases. Now something I'm also considering is I'd really like this to end in a GC clamp. So you guys can see if I extended this a couple bases further, it would go to a T and a G. I'm going to end right here at this G, right? So I'm going to add that T and the G to my sequence. So I'm just going to go right up into here, and I'm going to hit TG, and then calculate, right? So now I'm at 23 for a length, and I'm at 58.8. I mean, that's getting really close. Let's see what happens if I extend this just a little further and I go with another T and a G, which might be good or it might not. Let's see. Okay. I mean, it's at 61, and now I'm at a length of 25. I would actually probably go back and just finish it right there. I'm close to 60 degrees. It's not ideal. What I might actually go and do is say, like, okay, well, let's grab a part of the sequence that's further back, or maybe I'd go like a little bit further forward until I got something that's pretty close to 60 degrees. The closer you can get to 60 degrees, the better, right? So now let's just say that this is my primer, right, that I would actually be using, right? So it's right here. Now I have all of this. You see all of this room I have before we get to the actual part of the sequence that we care about. It's giving me a lot of room for this to be all that noisy stuff that gets sequenced. This is the part that I really care about. This has to be like a good clean read. So my forward primer has to come back here somewhere. Now my reverse primer doesn't really matter as much. The most Sanger sequencing can do is about 1,000 base pairs. So I would just start maybe designing right back here. Now reverse primers are a little bit more tricky, right? So I'm just going to grab somewhere around here and get like 20 bases, right? And I'm going to hit copy, and then I'm going to go back up to here, and I'm going to hit paste, right? So it starts with AAA, and you guys can see if I go back to my snap gene here, it thinks what I want is this 5 to 3 prime direction, this AAA. We know for a reverse primer that's not what I want. What I'm actually interested in is this sequence down here for the reverse primer. So what I'd like to see is something that goes G, C, C, A, G, A, right? That's what I would actually be typing in if I was going to order this. So there's a pretty nifty trick that you can do on here, and that's just the reverse complement. And you guys can see that's already filled out right here, right? So it's got G, C, C, A, G, A. Well, that's perfect. We've got G, C, C, A, G, A, right? So it's reading just the backwards or the reverse complement. So it's already making your reverse primer for you. Now, another cool thing that you can do is you can say, all right, I want to swap strands because I'd like to analyze that. And now it just jumps from this box right up to here. So now check it out. You can look at all of your criteria right down here. So GC content looks good at about 55%. This isn't even close to a high enough melting temperature. So I'm going to go grab a few more nucleotides. And then look at this. So we got a G and a C next. So let's see what happens if I add a G and a C. Right, so I have G and C. And then let's hit calculate. I'm up to 58.6. It's pretty good. Right, so there's my G and my C. Yeah, so I would go ahead and leave it there, right? And then that would be my reverse sequence. Now those are going to be my 2 primers, and I've pretty much accomplished all the goals that I want. I've got the length correct. My GC content is good. My melting temperature is pretty darn close to 60 degrees. If this was actually me and I was actually designing these primers, I would keep going until I got something that was very, very close to 60, and I would just kind of play around with this a little bit, right? But then your other criteria is met in that we've got something, the forward primer's way over here, our reverse primer's way over here, so now we know that our sequence is going to be a nice clean read, and this is where our SNP is at, and that's the thing that we want a clean read of. So next thing I would do is I'd write down my forward and my reverse primers, both from 5' to 3', submit those to me, I'll check them out, and hopefully those primers look good and we'll actually order those from IDT, and we'll do some sequencing with those primers. But I thought this was just a cool opportunity for you guys to actually design something in lab. Instead of me giving something to you, this is exactly what you would do if you were doing some sort of research project and you had to design some primers. So give it a try. It's a cool skill to try. And we'll check out and see who did the best, and we'll actually order those primers. Okay, thanks guys.